very kind, loving Christians who try to make the theory of evolution and the old age of the earth work out with the Bible. And there are some very sincere, well-meaning people on that side, which, who I love and respect, but I believe they are completely wrong, and I think it's very easy to show that the earth is very young. If you go by the Bible gives us a timeline showing us that the earth is about 6,000 years old. If you want to talk strictly science, there is no way the earth could be any older than 10,000 years. Now that I know is very different, and it was shocking when I first heard that after I started reading the Bible for the first time. But then when I looked into empirical science, I found that that is all that is available, is that the earth could be at most 10,000 years old. Now, the biblical timeline then says the next big event was 4,400 years ago. There was a worldwide flood in the days of Noah. And that was when Noah, his wife, his 3 sons, and their 3 wives got on a boat. The entire world flooded. There was a flood that covered the highest mountains all around the world. And that is the event that caused the majority of what we see in geology today, a worldwide catastrophe. Then the Bible tells us about 2,000 years ago, Christ was born, and about 33 years after that, he was crucified, and that single most documented death in the history of the world. Then here we are, present day, and we are in church talking about the Bible. I don't know where I am in my notes, so give me a second. Okay, here we go. So that brings us to our topic tonight, the origin of life. So the question we're asking is, where did we come from? After all, we're here. We're alive. I don't think any of us would argue that. Life exists all around us. The secular humanist requires that there's a naturalistic explanation for everything, an explanation without God. So, how did the first life appear? The current theory is that life emerged from chemistry. We're going to start by looking at what they are teaching our children in the public schools. And I hate to say this, but if for no other reason, we're going to do this just to have a good laugh, because the idea is absurd. Here we read, millions of years of torrential rains created great oceans. Swirling in the waters of the oceans is a bubbling broth of complex chemicals. Progress from a complex chemical soup to a living organism is very slow. So this is what is being taught in the textbooks in our schools, that you came from soup. Okay, remember kids, there is no God. You came from primordial soup. You can't make this stuff up. Now, here are the 6 different kinds of evolution. Notice that I labeled types 1 through 5 as religious beliefs. I do that because the first 5 definitions of evolution are outside of the scientific method. The origin of time, space, and matter. The origin of stars, planets, and the celestial bodies. Chemical evolution and the order of the higher elements. And what we're going to talk about today, organic evolution. All of these things cannot be tested, demonstrated, or replicated in a lab. They are therefore outside of the scientific method, which means that you cannot falsify these. That makes it a religious belief. It is something that is taken on faith. It is not taken because the evidence points us in that direction. Now, tonight we're talking about number 4, organic evolution, the origin of life from non-living material. Because after all, that's what we're dealing with. The earth was formed, there was non-living material, and then life had to appear. Which again, if you want to believe that, that's fine. I'm just saying get it out of the science class. So here we start out with a quote from Francis Crick. He discovered the DNA molecule with James Watson, and they received a Nobel Prize in 1962. He says about this subject, an honest man armed with all the knowledge available to us now could only state that in some sense the origin of life appears at the moment to be almost a miracle. So many are the conditions which would have had to have been satisfied to get it going. There are 2 great biological questions that the evolutionist has trouble answering. The first one is, how do you get new living forms with new structures, like wings and eyes, from life that already exists? This is the idea of you have a simple life form, and it's going to turn into a complicated life form over time. So how does the fish come out of the water and learn to breathe air? We've been told that reptiles turned into birds. Well, that's nice, and you can put pictures of them next to each other, but it doesn't describe how birds are warm-blooded and reptiles are cold-blooded. It doesn't describe how birds have a 4-chambered heart and reptiles have a 3-chambered heart. If you want to believe one turned into the other, that's fine, but we cannot duplicate that in a lab. We cannot replicate that. There is no evidence to show that. The second great question is, how did life originate on Earth in the first place? And that's what we're going to talk about tonight. Now, as far as the first question goes, the idea was really put forth by Charles Darwin in his book, The Origin of Species by Means of Natural Selection, or the Preservation of Favored Races in the Struggle for Life. Now, many of you might not have heard that long title, but that is the title of his first book. And to answer the question, yes, Charles Darwin was a terrible racist. If you don't believe me, get his second book. You won't find it in high schools or colleges, okay, because those thoughts and ideas are not allowed there. It's called The Descent of Man. He is insanely racist. But he came up with the idea that all life shares a common ancestor. You, your dog, your cat, and the banana all have, from the tree of life, go back down to share a common ancestor. Now, he wrote his book in 1859. In 1859, he didn't offer any explanations for how this happened. He certainly didn't offer any evidence to how this happened. And here we are in 2019, and the evidence still does not exist. Both the origin of life and the origin of the major groups of animals remains unknown. So let's get to the topic tonight. We're actually going to talk about biological evolution and macroevolution, how one type of animal turns into another. We're going to do that in a couple weeks from now. Tonight, we're going to talk about the origin of life. Now, if you go back a couple hundred years, you find out that in the early 1600s, you can find a recipe for mice. The Belgian-born chemist Jean-Baptiste von Helmont said, "Old dirty shirts placed in an open vessel of wheat grain will produce mice in about 21 days." Now, you can't make this stuff up, but this is historic. Look it up. Don't believe me. There are recipes for flies, how to make frogs. There are recipes for all of these things because the people didn't understand where they came from. Now, they even did experiments where they would crumple up a bunch of dirty rags and throw them in a corner with some old moldy bread, and they would watch it, and all of a sudden mice would appear, and they believed that's where mice came from. Now, these scientists didn't understand the concept of a control in an experiment, but that's what they believed. And once again, that's fine, but it was proven wrong. The idea of abiogenesis is now a theory, a former theory, now rejected. The definition is the generation of living from non-living matter. Spontaneous generation is the title. A former theory now rejected. Well, why is it rejected? Because of this fella. Louis Pasteur and Francisco Redi came up with the law of biogenesis. And this law states, very simply, that life only comes from living organisms through a process called reproduction. That's all we've ever seen. That's all we've ever demonstrated in a lab. It will never change. The law of biogenesis. So, nobody knew where the flies came from. A simple experiment showed us that they were produced from existing flies. The current theory was that they appeared spontaneously from old rotting meat. Now, we all agree that spontaneous generation is not true, right? I mean, we don't have to go over this. None of us believe this. It's not taught anywhere. It's been rejected. But do you know what they did? They just changed the name. They changed the name and said, oh, now it's chemical evolution. We get a bunch of chemicals together, the right chemicals. and you have those chemicals with the right amount of energy, and you have the right amount of time, and I'm going to add, abracadabra, life. And that is what is taught today. Again, we cannot test it, demonstrate it, see it in a lab. It is not a falsifiable theory, but that's what they believe. Alexander Oprin, a Russian scientist whose book, The Origin of Life, had a theory that supported an atheistic worldview that excluded God rather than one based on the evidence. Scientists who promote a spontaneous origin of life without a creator begin with the assumption that God does not exist. Now, good science doesn't start with any assumptions. Good science starts with a theory, and then the evidence leads you. After assuming God does not exist, then they devise an imaginative scenario whereby molecules came together to form a living being. Since the evolutionists have an explanation, they use it to insist that life can arise without God. It's circular reasoning at best. After this, we needed an experiment to help solidify this idea. And here enters Stanley Miller in 1952 with his experiment. I believe it was at the University of Chicago where this went on. Some believe that this was the closest a chemist ever came to producing life in a laboratory. And this experiment was a pathetic failure. Now, I should say, that doesn't speak poorly to Stanley Miller. Most experiments are a failure. That's what science is. And then you revise your theory and you try again. There's no shame in performing an experiment and finding out you were wrong. That is the majority of science. What is a shame is knowing what he produced and the failures of the experiment and still putting this in textbooks today in this town, in colleges and universities around the country, and saying that this is a great evidence that life could emerge from non-living material. That is dishonest at best. Before we get into the Miller experiment, I want to go over just a couple of terms to make sure we're all on the same page. So this here is a picture of a cell. This specific cell is a bacteria, and it's the one that gives humans pneumonia. The cell is often referred to as the simplest form of life. And whether you want to debate what life is and if a single cell counts, that's a discussion for another day. But today we'll say a single cell would be fine. That's good enough. We'll call that life. Simple enough. Now, every cell on planet Earth, without any known exception, has several characteristics that are shared. And not only the cell, but you have these same characteristics. All life have at least these three minimum characteristics. Number one, they have the ability to reproduce. Now, I understand that some people, some animals, some cells are unable to, but they have been equipped with the ability to reproduce. Number two, all organisms constantly sense changes in their surroundings and make response to those changes, and the communication between a cell or any living organism and its environment is called homeostasis. All living creatures have this. And number three, all organisms require energy to maintain the process of life. Living organisms must have the capacity to obtain and convert energy from their surroundings to grow and maintain themselves. This phenomenon is known in biology as metabolism. Every single living organism shares those three characteristics, down to the cell. Now, if we consider the cell to be the simplest living creature, great. How do we get one? Couple more terms before we answer that question. Number one, we have the atom. We talked about that, I believe, a couple weeks ago. We remember that, right? Periodic table of elements. hydrogen atom, one electron whirling around on the outside. Okay. We can chemically bond some of these together and make a molecule. You take 2 hydrogen, you take 1 oxygen, you got a molecule, specifically water. Okay, it's made up of atoms. Bonded together, we have molecules. After the molecule, we have amino acids. Now this and the next one are what are important for tonight. So we're going to talk a little bit about these 2 things. Living cells are made of special molecules. The building blocks of life are called amino acids. They form highly complex and orderly structures called proteins, cell membranes, and genes. A chain of amino acids bonded together can contain as few as 2 and as many as 3,000 amino acids. So you have one amino acid, then another bonds to it, then another bonds to it, then another bonds to it. When the sequence is correct, they fold up on themselves to make a 3-dimensional structure called a protein. The proteins are responsible for the structure of a cell, its chemical reactions, and they act as antibodies against bacteria and viruses that might attack the cell. There are about 500 naturally occurring amino acids in the world. But here's the neat thing. In every cell in your body, in the body of your dog, your cat, the banana, out of those 500 amino acids, only 20 are found in life. In every protein, in every cell on planet Earth. Much like the 26 letters of our alphabet are used to form every single word in the English language. So, if you think about it, you have the amino acid, makes the protein, makes the cell. The average protein has 300 amino acids linked together in order to make the protein. That would be like a sentence being formed with about 300 characters. So I counted one up. Okay? In the beginning, God created the heaven and the earth was without form and void, and darkness was upon the face of the deep, and the Spirit of God moved upon the face of the waters. And God said, "Let there be light," and there was light. Letters, spaces, punctuation, there are 248 characters in that long sentence, those couple verses. It takes about 300 amino acids, all in the right order, to make one protein. How many proteins are there in the simplest cell? 42 million. So the amino acid is like the letter in the English alphabet. The protein is like a pretty big long run-on sentence. And the cell, it's not a book. It's a volume of books. It's a volume of many, many, many books. Okay, and that's how we make cells. The amino acid forms the proteins, makes the cells. So, what's up with Stanley Miller's experiment? Did he create life or not? Anyone that has taken any science class in high school or college has seen that contraption. He circulated 4 ingredients through his contraption: methane, ammonia, hydrogen, and water. They were heated, and the gas passed through an electrical spark. Then it passed through a condensing chamber to turn it back into a liquid. That liquid was filtered out into a trap. Now, let's take a look at what the textbooks say when describing this experiment. And you tell me how good of a job he did. Scale of 1 to 10. 1 would be he blew up everyone in the room. 10 was he created life. Okay, let's see how well he did. The textbooks say he did add evidence to the theory that life could have started by itself on primitive Earth. Added evidence. Many important events occurred during the Archean era, the most important of which, according to scientists, was the evolution of life. Did you hear that? They nailed it down to certain number of years. The first living cells emerged between 4 billion and 3.8 billion years ago. There is no record of the event. It actually says that. I love that. His experiment produced the chemicals of life. And finally, the bottom of his flask was rich in amino acids. So the textbooks make it sound like he did pretty well. But they left a lot out. Like every single one of the problems. So problem number 1 in the Miller experiment. What ingredients were used and would they produce life? So one thing he did not use was oxygen. No oxygen was present. Now, why did he omit oxygen? He had to. It wasn't because of the evidence. It was because of the first ingredient right there, methane. You want to know what happens if methane passes through a spark and there's just a little bit of freestanding oxygen? Yeah, he would have blown up himself and everyone in the room. But that's not even the biggest problem. The problem is amino acids will bond more quickly to oxygen than they will to each other if there's freestanding oxygen. The amino acids will become oxidized and they will become worthless. Oxidation is what happens when you take one bite out of an apple and set it on a counter and leave it for several days, like my kids do a couple times a week. It turns brown. All the sugars rise to the surface of the apple. That's called oxidation. It's a chemical process. Oxygen actually destroys almost everything it touches. That's the reason he kept it out of the experiment. Nothing in our natural world suggests that Earth had an oxygen-free environment. He didn't do this because of the evidence. He did this because he had to if he wanted even a slim chance of success with his experiment. There is no scientific proof that Earth ever had an oxygen-free atmosphere, such as evolutionists require. Earth's oldest rocks contain evidence of being formed in an oxygen atmosphere. Evidence of free oxygen has been found in rocks older than the first living cells. That's from Geology magazine. Now, if there was no oxygen on Earth, then there would be no atmosphere. What is our ozone layer made up of? O3, oxygen. Without an ozone layer, the ultraviolet light from the sun would have destroyed all the ammonia present. The bigger problem would be any amino acids that were hit by ultraviolet light would have also been destroyed. Would not have been a good recipe for creating life. Problem number 2. Did Miller use chance or intelligent design? Now let me ask you this question. What was the purpose of his experiment? What was the goal? Because prior to 1952, we've already synthesized amino acids in labs. That wasn't new or surprising or impressive. That's already been done. The goal was to see if we could make the chemicals of life. Let's put out of our mind that he was going to make a living cell in a lab, okay? Let's just say the goal was to make the chemicals of life, the building blocks of life. Well, did he use naturalistic processes and chance, or did he use intelligent design? You see, the whole experiment was an example of intelligent design. He determined the gases to use. He determined the quantity of the gases. He determined when to generate the electrical spark. And then he made a trap to filter out the product. He didn't want it to go through the contraption a second time, because if it was heated up in the boiling flask, or if it went through the electrical spark a second time, any of the organic molecules he made would have been destroyed. So no matter what he produced, the only thing he would have proved is that to make organic molecules requires intelligent design. Problem number 3, and this is the nail in the coffin. What was the product of the experiment, and is it beneficial for life? Now, I have not read in one textbook, and I have a collection of them in my office. Every science textbook I can get my hands on from high schools and universities, I get and I go through and I highlight it and figure out what it says, and not one time have I ever seen them say what he actually produced. He did not produce a living cell. He did not even produce a single protein. And remember, the simplest cell has 42 million of those. He did produce some amino acids, and that is what made the headlines in the newspapers. But that's only half the story. Remember, we said there are 500 amino acids naturally occurring in the world, but only 20 are found in living cells, right? Well, there's another thing. Amino acids all come in two forms, left-handed and right-handed amino acids. They're like a mirror image of each other. Every single cell in your body, in your dog, your cat, and your banana, in all life, every single protein is made up of 100% left-handed amino acids. One right-handed amino acid and the protein is worthless. And nobody knows why. Not only that, Stanley Miller produced an even number of left-handed and right-handed amino acids. He did not produce the chemicals of life. But we know exactly what he produced, because the second you die, every cell in your body breaks down, and every left-handed amino acid converts into an equal number of left-handed and right-handed amino acids. Every living cell on planet Earth is the same. Stanley Miller produced the chemicals of death. They didn't put that in the textbooks. Every experiment that has ever been done ends up with a mixture of left-handed and right-handed amino acids, even when you start out with 100% left-handed amino acids. The tendency is always away from life and never towards life. Now, what else did he make? He made 85% tar, 13% carboxylic acid, and 2% amino acids. What he made was a vat of poison. They didn't put that in the textbooks either. Now, it takes more than the right ingredients and adding some energy to get life. If you don't believe me, there's an experiment that you can do at home in your own kitchen. Get yourself a frog and throw him in a blender. Turn that sucker on and let it whirl. About 30 seconds or a minute, you got frog soup. Now let me ask you, in that blender, is there every single ingredient required for life? Sure is. You do whatever you want to it. Electrocute it, microwave it, radiate it. That will never be a living organism again. Every ingredient's there in the right proportion. Never going to be life. Why is that? It's because the law of biogenesis. Do you know who invented and created all the laws that govern the universe? God did. In Genesis 2:7, we read, "And the Lord God formed man of the dust of the ground and breathed into his nostrils the breath of life, and man became a living soul." God created every living creature on earth, and from that day until this one, life produces living material. That's it. We only get life from life. It's the law of biogenesis. Scientists have been having so much trouble with this idea that they say terrestrial explanations are impotent and non-viable. Do you know where they're looking for life to have started now? They're looking for extraterrestrial answers. That doesn't help any. That just pushes the problem further away. But the evolutionary scientists have had so much trouble with their experiments, and the more we learn, the more we realize that there is something special to life, and it is nothing that we possess. We can't add it to the right ingredients. Now, we only got to talk about one or two more subjects, but I'm just going to say this. If you thought that you were getting out of here without listening to Patrick do some math, you are crazy. So we're going to go over the odds of producing life. Now, we talked about this in the past, but we have some new folks here, and who knows who's watching online. So we're going to go over the basics for odds. What are the odds of getting heads when you flip a coin once? 1 out of 2. We all agree? Okay. What are the odds of getting heads twice in a row? Well, you take 1 out of 2 and multiply it by 1 out of 2. You multiply the denominator. So 2 times 2 is 4. Getting heads 3 times in a row, the odds are 1 out of 8. Getting heads 8 times in a row, 1 out of 256. Now, remember, we need molecules, organic molecules called amino acids, to bond in the right order. One, then the next, then the next, then the next, then the next. What is the average number of amino acids in a protein? 300. They come in left-handed and right-handed. There are also 500 of them on Earth, and only 20 of them are used in life. And just like letters in a long run-on sentence, you can't just start mixing them up. It doesn't make any sense. It doesn't work. So what are the odds of getting heads 100 times in a row? Well, that number is 1 in 2 to the power of 100. Now, that number is so big that there's not a name for it, which is why we use scientific notation. But that number is a 2 with 100 zeros past it. To give you an idea, not talking odds now, just to give you an idea of the size of that number, if you wanted to flip a coin that many times, 2 to the power of 100, you'd have to flip that coin 1,000 times a second for over a quadrillion years. Okay? And the odds of getting heads 100 times in a row are 1 out of that number. And how many amino acids in a sequence for the average protein? 300. So what we have is the law of probability. And I will say this again and again and again and again. If you are a scientist or you are a mathematician and you believe that we came about by naturalistic processes, you have not done the math. The law of probability is 1 in 10 to the 50th. That's a 1 with 50 zeros past it. And that is essentially a law of impossibility. That event can never happen. In order to get a single protein, the odds are 1 in 10 to the power of 191. To get a single cell, the odds would be 1 in 10 to the power of 40,000. Now again, if you want to believe this, that's fine. That's fine. But get it out of science class. It is not scientific. Francis Crick was asked what the odds were. Again, don't believe Patrick. Remember, he believes the Bible. He can't be a scientist. He can't be trusted. Francis Crick is not is not a Christian. Doesn't believe the Bible, not a creationist. He did win a Nobel Prize for physiology or medicine by discovering the DNA molecule. And this is what he said. If a particular amino acid sequence was selected by chance, how rare an event would this be? Suppose the chain is about 200 amino acids long. This is, if anything, rather less than the average length of the proteins of all types. Since we have just 20 possibilities at each place, the number of possibilities is 20 multiplied by itself some 200 times. This is conveniently written 20 to the 200th power and is approximately equal to 10 to the 260th power. This number is quite beyond our everyday comprehension. The great majority of sequences can never have been synthesized at all at any time. Now, the amino acids are linked together in a long chain. Only 20 of them used for life, all left-handed, one, then the next, then the next, then the next, then the next. And then they fold in on each other to make a three-dimensional structure called a protein, which has various uses in a cell. If the amino acids are sequenced correctly, then the chain will fold up into a functioning protein. Proteins are arranged with their amino acids in such a way that the amino acids collapse on each other in an architecture that is pre-programmed by the order of the amino acids. So it folds into a certain structure, and that structure can do a certain function. So all proteins in the cell have a certain 3D pattern that is based on the arrangement of the amino acids in the chain. Now, the arrangement is critical. Here we have hemoglobin, the blood's oxygen-carrying protein. It has two chains containing a total of 287 amino acids. If you substitute, so we're going to remove one amino acid and put the wrong one in its place. So if you substitute the amino acid valine for glutamic acid in the number 6 position of the beta chain, the result is an incurable condition called sickle cell anemia. Even though the other 286 amino acids are assembled perfectly. This shows how flawlessly a protein structure must be. Most of the time, one wrong amino acid and the protein is just worthless. It cannot be used by the cell. But if it ever is used by the cell, it is always a mutation that is harmful to the cell. So, here's the question. How does your body know when it's producing more cells out of the amino acids, out of the proteins? How does your body know what to make? Where are the blueprints? Okay, where's the diagram of how to build this thing? Well, that is called your DNA. Deoxyribonucleic acid, a very complex molecule. And this molecule is found in almost every single cell in your body. The structure is called the double helix. It twists up, and as it does, it compresses on itself into the DNA molecule. This stores all the information that makes up you. Every cell, okay, all of your genes, the color of your eyes and your hair, and whether your hair is straight or curly, every detail about you. This is the blueprint. And it's contained in trillions and trillions and trillions of cells in your body. Bill Gates said, DNA is like a software program, but it's much more complex than anything we've been able to design. Now, how much information is in one DNA molecule in one of your cells? Well, if you took all the information in a DNA molecule and typed it out in a Microsoft Word document, just a white page with black text on it, it would be just over 2 million pages. If you stacked those pages flat in one long, tall stack of paper, it would come out to 667 feet tall. The Washington Monument is 500 feet tall. That's the amount of information contained in one cell of your body. And that's contained in every cell, in every animal, every plant, every person. Now, keep in mind, that it's not contained in your hair cells and mature red blood cells. I understand there are some cells that don't contain DNA. Okay, that's not the point. If that's what you're taking away, you're missing the point. All the information is in the nucleus of almost every cell in your body. Here's a statement. This is a true or false statement. The simplest operation of a cell is still more complex and complicated than an operation of a computer. True or false? You know where that's found? That is a question on a 9th grade biology test. Francis Crick said biologists must constantly keep in mind that what they see was not designed, but evolved. Why do you need to constantly keep that in mind? Well, because what you see every day through the microscope or the telescope screams that there is a creator. God made sure that even the casual study of the natural world will always point to him. There is no way around it. That's why I said, you have to remember, we evolved. Keep telling yourself, we evolved, we evolved, we evolved. There's no God, there's no creator, there's no designer, we evolved. Biologists have to keep telling themselves that. Why? Because everything you see in your studies points to an intelligent designer. Do you know that you are capable of determining something that was designed? If you take a walk through the woods, taking a hike, and you come across a laptop computer, do you think to yourself, this is amazing. This must have taken millions of years to form. Where did all the metal and silicon and silver and the glass for the screen, this must have taken millions of years of naturalistic processes to come up with this laptop. This is remarkable. Nobody thinks that. Because you're able to identify something that was designed. What you think is some knucklehead left their laptop in the trail. But for some reason, in science class, they tell you if it's biological, it could not be designed. Even though every scientist, far smarter than Patrick, says that biological systems are more complex than anything that we've ever invented, you still have to remember it was not designed, it evolved. That's what the kids are told. And they're told that so much that they start to believe it. And if you run into someone that looks at a leaf and thinks the cell in the leaf that can take visible sunlight and turn it into usable energy through the process of photosynthesis, if you think that that came about by accident, you can call that person a fool. Now, I don't think you should. I don't think that's polite, but it's accurate. That is absurd. Do you know that we are still trying to design solar panels that will do half of what the cells in the trees can do? And they're expensive. That's it for the origin of life. I have one more question for you and we'll be done. I don't know what time it is, but I feel like we got it in under the hour. Last question. Are you 100% sure that if you were to die today, you would go to heaven when you die? There's not a right or wrong answer to this, but there is an honest one. And I can tell you that the Bible says that you can know for sure. Not wonder, not hope, but know. Whosoever shall call upon the name of the Lord shall be saved. You don't have to hope you're saved. You don't have to wonder if you're saved. You can know for sure. God wants you to know. If you die today, you'd go to heaven when you die. And if there is anyone here in the room tonight or out in Facebook land and YouTube world that's not sure, I would ask you, please, let me take the Bible, let me take 5 or 10 minutes and show you how you can know for sure that heaven is your home. My name is Patrick Hayes. You can contact me at BibleStudyOnTheFarm@gmail.com. And please follow us on Facebook.com/BibleStudyOnTheFarm. You can find all of our videos there. And with that, we're going to close with a word of prayer, and we'll be dismissed. God, we love you greatly, and thank you, Lord. Just thank you that we live in America and we still have the freedom to come together and talk about the Bible and talk about you. We're grateful for that. Grateful for all the folks that showed up. I hope that we had a good time. We learned something about you, and